Welcome everyone back to Anatomy and Physiology 2, and in this discussion we're going to talk about metabolism. So clearly this chapter goes right along with what we just discussed previously in digestion, and that we talked about the mechanical aspect of digestion, the chemical aspect of digestion, absorption, and excretion. So now we're going to talk about sort of the chemistry behind all of that and what is metabolism. So a lot of times when you think of metabolism, you just think about one end of metabolism, and I'm just going to kind of scroll through these learning objectives, which are all posted on Blackboard. You just think about the part of metabolism where you break things down, but metabolism also has anabolic properties where things are going to be built up. So the amino acids that we get from the breakdown of the proteins that we eat from our diet, those can be built into various different proteins for your hair, for your hemoglobin, for what we consider structural proteins, functional proteins, enzymes, for various different things. So what we'll do in this chapter is that we're going to talk about both aspects of it. A majority of the time will be spent talking about the catabolic aspect of metabolism, or the part of metabolism where we break macromolecules down and get energy out of it. But we will talk about the anabolic properties of metabolism too. So metabolic activity, cells break down organic molecules, and when we talk about organic molecules, we're talking about the Fab4, carbohydrates, proteins, lipids, and even nucleic acids to obtain energy. That is a catabolic process. We break things down, break the bonds in those macromolecules, carbs, lipids, proteins, nucleic acids, and get energy out of it. So we use that to generate ATP. Most of this energy production, most of this process, the most efficient process is cellular respiration, aerobic cellular respiration, and that happens in your mitochondria. Now for those of you all that are taking micro, or have recently taken micro, or you've recently taken an introductory biology course, you're going to be pretty familiar with what we're talking about in this chapter. So a few definitions here. Energetics, the flow of energy as it changes from one form to another. So in the apple that you plan on eating for lunch, the flow of energy, the bonds in that apple will eventually be broken down through your mechanical and chemical digestion, through your absorption. You will then take that, let's see, apples have carbs in them, so you'll take those carbohydrates, break them down into individual glucose molecules, and you'll get energy out of that. So we're changing it from a potential energy, the apple just sitting there, into an active energy or kinetic energy, an energy in motion that you will use. So even though you don't actually see the apple doing anything as it's sitting on your desk, know that there is potential energy in that. And we know that because we can eat the apple and feel more energized. We feel like we have more energy. Metabolism, as I said very early in this discussion, is the sum total of all the chemical reactions that happen in your organism as it relates to breaking down macromolecules, the Fab4, and then building up the monomers, amino acids for proteins, glucose for carbs, nucleic acids, the nitrogenous bases for nucleic acids, and the lipids and the fatty acid, the glycerol and the fatty acid tail for lipids, building them up into something else. So cellular metabolism includes all those reactions in the cell, and that's the energy that's needed to maintain homeostasis and also to build different types of proteins and carbs and give energy for the necessary functions of your cells. So here is a pretty picture of a cell. It's just kind of a nondescript cell that we have here. And that we have both the catabolism, the breakdown of the foods, and then we have the anabolism. So the results of anabolism is that we will be able to perform maintenance and repairs, growth, secretion, stored nutrient reserves, so from those building blocks from that nutrient pool. Catabolism is going to break things down and put those monomers into that nutrient pool. We'll also get energy out of that process, and the organic molecules that are coming into the nutrient pool are these amino acids that build proteins, lipids that will build, or glycerols and fatty acid tails that will build lipids, and these simple sugars like glucose that will build complex sugars such as carbohydrates. Anaerobic metabolism happens in the cytoplasm, so that's really glycolysis, and we do get a little energy out of that, but we don't get nearly as much as we do from aerobic metabolism, which happens in the mitochondria. So we get some of that, about 40% of the energy that comes in from that nutrient pool, from those organic molecules of proteins, carbs, and lipids. 40% of that energy we use to power things that happen in the cell: locomotion, contraction, intracellular transport, cytokinesis, endocytosis, exocytosis, all of the stuff your cell needs to do, so that it requires energy for. And then about 60% of that is going to be released off as heat. So I'm kind of going through this a little faster than I would if this is the first time that you've seen this. If it is the first time that you've seen this or had any discussion in a talk about cellular metabolism or in cellular respiration, you will want to go back and read the information in your book very carefully. And I also have some videos on YouTube in microbiology that will talk at great length about this whole process of energetics. So if you're not familiar with them, there are also fabulous other YouTube videos on there from Khan Academy that will give a more in-depth discussion. But I'm coming to you all from the point of view that you've seen this material before. So as we said before, there are two parts of metabolism. It's an integrated process where we have the breakdown part of it, catabolism, and we have the buildup part of it, anabolism. Catabolism is going to give you energy, and it breaks down organic substances. Remember the organic substances we're talking about: carbs, proteins, lipids. Anabolism is the synthesis or the building of new organic molecules, and it's going to form new chemical bonds. So instead of releasing energy in an anabolic reaction, it's going to actually absorb or require energy. So the organic compounds that we're primarily talking about, keep in mind that nucleic acids are also an organic compound, and nucleic acids, there is some energy that you can get from breaking those down, and you incorporate those nitrogenous bases of A's, T's, C's, and G's into your own DNA. But we don't really talk about them as much in metabolism, but I do want you to recognize that they are organic compounds as well. The organic compounds that we're mostly concerned with that will give us the most energy are carbohydrates, glycogen, triglycerides, lipids, and proteins. So glycogen is the most important organic compound for storage in the form of carbohydrates. It's a chain of glucose, and they're usually branched off. Glycogen is stored in your muscles and in your liver. Triglycerides are also storage. The most abundant storage molecule are lipids, and they're primarily in the form of fatty acids, where we have a glycerol head and 3 fatty acid tails, hence the name triglycerides. And then proteins are the most abundant organic compound in your body, period. Proteins can do all sorts of things. They are enzymes. Every enzyme you ever encounter is going to be a protein. So enzymes are proteins. You have structural proteins that make up your hair. You have contractile proteins that are in your muscles that will allow, remember, actin and myosin, that will allow for contraction to take place. You also have proteins that are meant to do various different things. So proteins perform many vital cellular functions, from enzymatic activity to contraction to being structural components that make up the cytoskeleton, make up the flagella, your hair, keratin in your hair, the keratinization of your skin cells, protein. So there's lots and lots of jobs of protein. If you go into any organ system, there is a clear discussion of the various different proteins that are responsible for the proper functioning of that organ system. So, looking at the nutrient use in metabolism, the red arrows are the catabolic pathways. This is where we're breaking these larger macromolecules down and getting energy out of them. And then the blue arrows are the anabolic pathways, where we're taking the smaller parts of it, amino acids for proteins, glucose for glycogen, glycerol and fatty acid tails for triglycerides, and we're building them up into other different things. So if we follow these arrows here, what we're really looking at as far as the catabolic aspect of it is that we're really just having a very truncated roadmap of cellular respiration, where the first step is that we take these nutrient pools And they're eventually going to go from being whatever it is they started off with to being, so whether it's fatty acid, a glucose, or amino acid, to be converted into a 3-carbon chain. That first step is glycolysis. Notice that if this whole block is a cell, that this first step isn't happening inside the mitochondria. It's happening in the cytosol. Glycolysis does not have to happen in the mitochondria in eukaryotic cells. It happens in the cytoplasm or the cytosol. Then that 3-carbon chain can further be converted into 2-carbon chains. So that's a prep step for step number 2, that one there, the Krebs cycle or the citric acid cycle. So Krebs cycle, citric acid cycle, they're the exact same thing, just 2 different terms for it. Now notice, amino acids can either go into this process with it being converted to 3-carbon chains, or, depending on what the amino acid is, and what kinds of things have chemically happened to that amino acid, it can also go into this prep step to be this 2-carbon chain, which we'll call acetyl-coenzyme A. The second step is that 2-carbon chain needs to go into the citric acid cycle, aka the Krebs cycle. From that process, we're going to get energy, just like we got energy out of step 1. We get ATP out of glycolysis. We get ATP out of the Krebs cycle. We're also going to have this byproduct of CO2, which will leave out of the cell. And then we're going to have these coenzymes, these electron carriers of NADH and FADH2 will go to the electron transport chain, which is the third step of the process of cellular respiration, where we'll have a tremendous amount of energy come out. And in order to facilitate this process, oxygen has to come into the electron transport chain to act as a final electron acceptor. And then we're also going to, at the end of the process, lose water. As I said before, this is just a truncated visual of really cellular respiration, but it's looking at it from a slightly different aspect, in that we're looking at how these different nutrients are used and where they can go into one of the 3 steps of glycolysis, the citric acid cycle, and the electron transport chain, also known as oxidative phosphorylation. And as I said before, a whole in-depth discussion of this you can find on various videos if you don't remember it. So here is step 2, the citric acid cycle. So remember we talked about 3-carbon sugar. So I'm going to go back and just show you this picture here. We have that 3-carbon chain. The name for that 3-carbon chain is called pyruvate. Pyruvate comes out of glycolysis, or out of step 1, which happened in the cytoplasm. In order for pyruvate to get into the Krebs cycle, it has to be groomed. So there are some things that need to happen to it. One of the things that needs to happen is that it has to lose carbon dioxide. So it's going to lose carbon dioxide out of it. Everything that's circled in green on this slide, these are products that are going to be products of the Krebs cycle. So things that come out of the cycle. Anything that's circled in purple here, those are things that go into the cycle. So before pyruvate can go into the cycle, a couple things have to happen. It has to lose CO2. In the process of losing CO2, it's also going to lose some, because it's a 3-carbon sugar and we've lost one of the carbons and an oxygen off of there, we're inevitably also going to lose hydrogens. And the hydrogen is going to be picked up by NAD, which is one of these electron carriers. Every time you see NAD or FADH, they go to one spot and one spot only, the electron transport chain. So in the process of losing CO2 and grooming itself to get into the Krebs cycle, we're also going to lose hydrogens that are picked up by NADH. Even though now we're down to a 2-carbon sugar, we still can't get into the Krebs cycle. We're going to have to be escorted in, escorted is a better word for it, escorted into the cycle by coenzyme A. So now that we've lost CO2, lost hydrogen, given that to NADH, which goes to the electron transport chain, hooked up with coenzyme A, we're no longer called pyruvate. We're called acetyl-coenzyme A. Once we get into the Krebs cycle, we no longer need that coenzyme A. We just have the acetyl group that goes in. So it's acetyl-coenzyme A that goes into the Krebs cycle. And as we go through these series of enzymatically timed events, we lose more carbon dioxide, we lose more NADH. Carbon dioxide exhaled off. And we talked all about that process in the respiratory system on the 3 different ways that we can get CO2 back to the lungs, etc. NADH, one stop that it's making, the electron transport chain. Notice all of these pink arrows that come from NADH and FADH2, they all go to one place, the electron transport chain. Then we lose more carbon dioxide, more NADH. We also are going to make ATP or energy. We'll have more NADH that's made. We'll also have FADH2 that's made. And remember, all of the things that are circled in green are products of the Krebs cycle or the citric acid cycle, and all of the NADHs and FADH2s, they're all going to go to the electron transport chain. Energy is going to be used for whatever. Now, because we have two molecules of pyruvate that go into the citric acid cycle, this cycle turns twice. So as a result, we're going to get 2 ATP out of this, one for each turn. And then this picture here is just more of a more involved in looking at the structure of each of these intermediates. And any one of these intermediates, isocitrate, alpha-ketocliteric acid, any of them can be converted into various different macromolecules. They can be converted primarily for proteins, different types of proteins. So we're looking at the catabolic properties of this cycle, but there are anabolic properties where each of these individual intermediates that we have here can be converted into various different organic compounds. If we put energy into them, we can make different things like proteins, lipids, and carbohydrates if we put energy in and convert them. Succinyl coenzyme A and succinic acid are just normal byproducts that come out of the citric acid cycle. But if you have too much of them, it can be toxic to you. So you could totally kill somebody with succinic acid. However, if their succinic acids are really high and the police looked at you as a suspect for a reason to kill them, and they have puncture wounds and there's various other things, and you will still go to jail. But your body does make a level of succinic acid. So the last step of cellular respiration is what we call the electron transport chain. So we referred to it before, and we said all of those NADHs and FADH2s go to the electron transport chain where they're carrying hydrogens and electrons. We can also call this process oxidative phosphorylation. So oxidative phosphorylation happens in the mitochondria, and it's where we're going to generate the bulk of our ATP. More than 90% of the ATP used by the body is produced through oxidative phosphorylation. So you can see why it's a more efficient means of getting energy than anaerobic or glycolysis. For oxidative phosphorylation to work, it has to have the electron carriers, NADH and FADH2, drop off the electrons and the hydrogens, and it also has to have oxygen. So with that oxygen, it's able to pull or drive the electron transport chain, and then oxygen acts as the final electron acceptor, where those electrons are moved from one proton pump to the next. Oxygen's kind of driving that whole force along, and as the final electron hooks up with the oxygen, the hydrogens that come back in that have been dropped off by FADH and NADH are going to be attracted to that oxygen and make water. So you're probably very familiar with this formula, C6H12O6 plus 6 molecules of oxygen give you 6 molecules of carbon dioxide and 6 molecules of water. Well, this is just a very generic formula for cellular respiration. And then this is for carbohydrate metabolism. So we have sugar plus oxygen. Oxygen goes into the electron transport chain. Sugar starts off in glycolysis, gets converted to pyruvic acid, and then converted to acetyl coenzyme A, which then further gets broken down and gives you energy out of it. From that conversion of pyruvic to acetyl coenzyme A, you have carbon dioxide that comes out of it. At the end of the electron transport chain, you have water. So we know where each of these compounds comes from. So to really understand this whole process, we must recognize that cellular respiration or aerobic respiration is really nothing more than a giant series of redox reactions where we have oxidation and reduction that are happening in several different places. Oxidation is termed as the loss of electrons, and reduction is the gain of the gain of electrons. And these reactions are always paired with one another. So where one molecule is losing an electron and hydrogens, there's another molecule that's going to gain those electrons and hydrogens. And that's why we just generically call them redox reactions. reactions. An easy way to remember who goes where and what goes where is oil rig. Oxidation is the loss of electrons. Reduction is the gain of electrons. Now, at first blush, it may seem like that's really backwards. Why is reduction gaining anything? But you have to keep in mind that electrons are negatively charged. So if you continue to gain these negatively charged entities, your overall charge, if you're positive, is going to be more negative because you're gaining negative things. So oxidative phosphorylation, or the electron transport chain, also happens in the mitochondria, as we saw with the Krebs cycle, or the citric acid cycle, happens in the mitochondria. It happens on the inner membrane of the mitochondria, and here are some of the players in all of this. The purple jobbies are the proton pumps that are going to transfer electrons from one proton pump to another. We have oxygen as our final electron acceptor. We also have the hydrogens that have been dropped off by NADH and FADH2, and the purple proton pumps, not only do they transfer electrons, but they're also going to pump out hydrogens. So when NADH and FADH2 that come from glycolysis, the first step, and the citric acid cycle, the second step, and even the prep step from the citric acid cycle, where we had to group the pyruvate, they're going to be dropped off at the electron transport chain, or for oxidative phosphorylation. The hydrogens have a different destiny than the electrons. Electrons will be transferred from one of these proton pumps to another, with oxygen kind of pulling this whole process, and so oxygen is the final electron acceptor. The electrons that are energizing these pumps will pump out the hydrogens. And those hydrogens that get pumped out, they don't want to stay out there. They've created what we call a chemiosmotic gradient, where there are more hydrogens in that inner membrane space than they are on the inside. So as a result, those hydrogens want to come back in to balance things out. They can't go back through the pumps that pump them out. They can't just go through that phospholipid bilayer of the inner membrane. They can only go through this enzyme called ATP synthase. So all of these hydrogens go through ATP synthase, and as they come back in, it's going to turn this enzyme, ATP synthase, so that ADP becomes phosphorylated, which means that it's going to have a phosphate group attached to it. ADP becomes phosphorylated and becomes ATP. When these hydrogens come back in, they're automatically going to be attracted to this oxygen because it now has extra electrons. These hydrogen ions, hydrogen has an atomic number of 1, which means it has 1 proton, 1 electron. If it is an ion, that means that it's a positive ion, a proton, it means it lost its electron. So hydrogens, all of these hydrogen ions have lost their electron. Oxygen, because it's the final electron acceptor, now has excess electrons. So the hydrogens are attracted to it, and as a result, we get water. So we get water and energy that come out of the electron transport system, also known as oxidative phosphorylation step. So now we've talked about all of those things. We've seen pictures of all of those steps. Now we're going to look at them in like the wordy form of it, so like the actual text of it all. First step in carbohydrate metabolism is glycolysis. One glucose molecule, which is a 6-carbon sugar, is broken down into 2 molecules of pyruvic acid. As a result, what comes out of glycolysis are 2 molecules of pyruvic acid, an electron carrier, NADH, and we're also going to get 2 molecules of ATP. Don't really worry about this whole transition phase sort of thing. That's why I've kind of crossed it out, because it sort of takes away from what I really need you all to understand. Those 2 molecules of pyruvic acid then go into the Krebs cycle. So since we have 2 molecules of pyruvic acid, the cycle turns twice, 1 for each molecule, and then we're left with carbon dioxide out of the Krebs, and the citric acid cycle, same thing. We're left with carbon dioxide, NADH, FADH2, and we get 2 molecules of ATP, 1 for each turn. All those NADHs and FADH2s, they're going to go to the electron transport chain. Each of the 8 NADH molecules will produce 3 ATP and water. Each of the 2 FADH molecules will produce 2 ATP and water, which gives us a total yield for carbohydrate metabolism of about 28 ATP. Now, if you've seen this before, which I'm pretty sure most of you have, you probably recognize that number changes. When microbiology, or when we talked about it in the muscle, I think we said there were 34 ATP that were produced. When we talked about it in microbiology, I think we said 36. So I just want you to remember it as a range, and there are various different reasons as to why this range is different. So between 32 and 38 ATP total are made from the whole process of cellular respiration. So as we said before, citric acid cycle breaks down those two pyruvate molecules, grooms them so that they can get into the citric acid cycle. We make 2 ATP, 1 for each turn of the cycle. Each of those hydrogens that have been lost go to the electron carriers, NADH and FADH2, and then those electron carriers provide the hydrogens and the electrons for the electron transport system or chain. Summary of ATP production, cell gains about 36 molecules of ATP. Really, we get 2 for sure from glycolysis. We get 2 from the citric acid cycle, and then we get about 28 or so from the electron transport chain, which gives you a total of about 36. But remember, I just wanted you all to remember this range. And that would be the range I would, if it's a multiple choice question, that I would give you for your exam. So a little bit more terminology here. Glycogenesis is the formation of glycogen from glucose. It's a slow process, and it requires a high energy compound in order for this to take place. Remember, glycogen is stored in your liver and also in your muscles. Glycogenolysis is the opposite of glycogenesis. Glycogenesis is storing up or building glycogen. Glycogenolysis is the breakdown of glycogen. This process happens quickly. It's just one enzymatic step when we want to break this down. If our blood sugar is very low, if we need to have glucose more readily available for whatever energetic activity that it is, then glycogenolysis will take place. So here we have glycogenolysis, where the glycogen is broken down, is represented by the pink arrow, goes through the process of glycolysis, and we have these different intermediates that come out of here, and we have ATP that comes out. Glycogenesis, more of an uphill anabolic sort of process or synthesis, where we can take any of these intermediates, combine them together to make this 6-carbon sugar of glucose, and then rearrange it to form this complex carbohydrate of glycogen that's stored in your muscle and your liver. Glycogenolysis happens much faster than glycogenesis. So lipid metabolism, keep in mind that you don't just consume carbohydrates, and that's really all we've talked about so far. You also consume lipids and proteins. Lipid metabolism is called lipolysis. It's the breakdown of lipids into pieces that can be converted into pyruvic acid, that 3-carbon sugar, to go through the last part of glycolysis, or they can be converted into molecules, any of those intermediates, to go into the citric acid cycle, or that second step. Hydrolysis is a process by which the triglyceride is split into its 2 components, the glycerol head and the 3 fatty acid tails. Enzymes in your cytoplasm will convert that glycerol into pyruvic acid, and it's that pyruvic acid that gets groomed and goes into the citric acid cycle. And then there are a different set of enzymes that will convert those 3 fatty acid tails of the triglyceride into acetyl-coenzyme A, and we call that process beta oxidation. And remember, it's this acetyl-coenzyme A that has been groomed to go into the citric acid cycle. So here is a pretty picture of all of this. We have our triglycerides get into the cell through endocytosis. A lysosomal enzyme will break down the triglyceride into the glycerol head and 3 fatty acid tails. The glycerol head will go through that last little portion of glycolysis, where it's converted into pyruvate, and then that pyruvate has to be groomed. Same things we saw in carbohydrate metabolism. When we get into the Krebs cycle, we lose CO2, We hook up with coenzyme A. Now it's acetyl-coenzyme A. We go through this process of the citric acid cycle, and every time it turns, we get 1 ATP out of it, NADH and FADH2, then go to the electron transport chain, and we have energy out of it. Bada boom, bada bing. So that part we've seen. We then also have the other portion of the fatty acid, the triglyceride, the 3 fatty acid tails. They will be converted so that they can turn into acetyl-coenzyme A. And what we have here is that the fatty acids get absorbed into the mitochondria because the citric acid cycle is in the mitochondria. And we can take just one portion of it. We don't have to take the whole thing by itself. We can just eat off little tiny portions of it where we have our carbonyl group and we have coenzyme A hooks up with this hydroxyl group over here. And or not the hydroxyl group, but it hooks up with the methyl groups, and then the hydroxyl group is going to be released off of there, and the hydrogens will be picked up by NADH and FADH2. So we have these small portions. Notice that only this purple part are we actually working with, and we can just bite off small chunks of this fatty acid tail. So the enzymatic reaction breaks off the first 2 carbons as acetyl-coenzyme A leaves the shorter fatty acid bound to the second molecule of coenzyme A. So we can just, as I said before, just bite off very small chunks. So as a result, lipids will give you a ton of energy simply because that fatty acid tail is so long that we can continue to have these acetyl-coenzyme A molecules go into the citric acid cycle, continually have these NADH molecules and FADH molecules go into this cycle to make energy. For each 2-carbon fragment that's removed from a fatty acid, you get 12 ATP from acetyl-coenzyme A in the citric acid cycle and 5 ATP. As a result, a cell can get 144 ATP from breaking down just one 18-carbon fatty acid molecule. Because of that, fatty acid breakdown gives you 1.5 times the energy of glucose breakdown. So the whole discussion behind like low-carb diets and being higher on fats and proteins, fats do give you more energy. They give you a significant amount of energy as a result of their breakdown because of the way they're chemically structured. Lipogenesis or lipid synthesis, non-essential fats and steroids are examples of other lipids. Believe it or not, cholesterol, estrogen, testosterone are in the lipids family. They're steroids. They can be synthesized from acetyl-coenzyme A. You have what we consider essential fatty acids and non-essential fatty acids. Essential fatty acids are those that your body cannot produce, so you have to consume them. They're unsaturated fats, which means that they have double bonds that cause kinks in the chain so that they're not so stiff. Or another way to think about an unsaturated fat is like an oil. It's not a solid at room temperature like butter. Linoleic acid and linoleic acid are 2 examples of essential fatty acids. You get those from plants in your diet, so your veggies and your fruits. Non-essential fatty acids are those that your body can make. Free fatty acids are important as an energy source, and your body uses those in periods of starvation or when your glucose supplies are limited. Your liver cells, cardiac muscle cells, skeletal muscle cells, and various other very energetically needy cells can metabolize these free fatty acids for energy. Lipoproteins are lipid protein complexes. They're large, insoluble glycerides and cholesterol. There are 5 classes of these lipoproteins that we have here. The chylomicrons, the very low density lipoproteins, the intermediate density lipoproteins, LDLs, low density, and HDLs, the high density. For lipid transport and utilization, a mycel is basically where we have this lipid that's bound or it's been broken apart by the bile, so that's why it's kind of green colored. It's been emulsified by the bile from the liver. We have this mycel. And the micelles are absorbed in the intestinal mucosa where they're converted to this chylomicrons, which was one of those 5 different types of lipoproteins that we talked about over here. So as they're absorbed in, the monoglycerides and fatty acids, triglycerides and other fatty acids will be broken down via exocytosis. The intestinal cells will then secrete the chylomicrons, which are absorbed by lacteals, and from the lacteals, the chylomicrons are processed within the lymphatic vessels into the thoracic duct and are then distributed through various places throughout your body. Mycoprotein lipase the capillary epithelium breaks down the chylomicron and releases the fatty acids and the monoglycerides into the interstitial fluid so that a resting muscle cell can absorb the fatty acids and then further break them down for ATP production or for storage as glycogen. Adipocytes are going to absorb the fatty acids and then use these to store the lipids in the form of a triglyceride. Your liver can absorb the chylomicrons and recycle to cholesterol, producing these low-density lipoproteins. Low-density lipoproteins leave the liver and they go into the bloodstream. The LDL is absorbed. Those peripheral cells outside of the bloodstream are going to absorb that low-density lipoprotein and are further broken down in the cell by the lysosomes. And then what we do is after we break down that LDL, we can use that for membranes, hormones, or various other materials. The byproduct is going to be released, so the cells extract the cholesterol from the LDL and they use it, and then what byproduct is left over, that unused cholesterol leaves, and it binds to the high-density lipoprotein. High-density lipoprotein converts cholesterol back to the liver for more recycling. So then the cholesterol is extracted and we go through this whole process again. Low cholesterol would be these what's extracted and not actually used, and it goes back into the bloodstream. So the last type of metabolism we'll talk about is protein metabolism. The most energy you get are from lipids. Second highest are carbohydrates. If no other energy source is available, then the mitochondria can generate energy by breaking down amino acids in the citric acid cycle or from protein catabolism. Not all of the amino acids, there are 20 different amino acids, can enter enter the citric acid cycle at the same point. So how many ATP you get is going to change. It varies. So for amino acid catabolism to take place, one of the first things that we have to do is remove the amino group by transamination or deamination. Either we move it or we remove it off completely. Requires vitamin B6 in order for this process to take place. Transamination is going to attach an amino group of an amino acid to a keto acid, and that converts, and keto acid can be converted into amino acids, which will then be available for protein synthesis. Deamination is going to prepare the amino acid to be broken down completely in the citric acid cycle, removes the amino group completely and a hydrogen atom, and that reaction is going to give you ammonia, which your body, we'll talk about that in the next chapter, urinary, your body will filter out. Ammonia ions are very toxic even if they're in low concentration. Your liver cells, which is the main location of this process of deamination and removing that amino group, have enzymes that can use ammonia ions to synthesize urea, which is excreted in the urine. So remember, we'll talk about that more in the urinary chapter, that process of getting rid of the urea. The urea cycle is a process that produces urea. So as I said before, when we use up all of our lipids and our glucose reserves, the liver cells can break down internal proteins, absorb additional amino acids from the blood. Those amino acids will be deaminated. Carbon chains are broken down to make ATP. But keep in mind that this is a last-ditch effort to use these proteins if we don't have enough lipid or glucose. So there are three factors against protein catabolism because, well, lipids and carbohydrates are just more efficient to break down and you get more energy out of them. But other factors against it is that proteins are kind of hard to break down. They're difficult to break apart than complex carbohydrates or lipids. You're also going to get that byproduct of those ammonia ions, which are toxic to the cells, and we have to go through that whole process of the urea cycle, converting them into the urea. And then 3, we need proteins for lots of structural and functional components of the cell. So we really don't, we need proteins for so many other things that breaking them down for energy really should just be a last-ditch effort. The body makes half of the amino acids, synthesizes half of the amino acids needed to build proteins. We call those non-essential amino acids. Amino acids that are made by the body on demand. It requires a process called amination. So deamination is the removal of an amino group, and amination is the addition of an amino group. It's NH4 is what we call that. There are 10 essential amino acids, and neither are 8 of them are not synthesized: isoleucine, leucine, lysine, threonine, tryptophan, phenylalanine, valine, and methionine. And then 2 are insufficiently synthesized: arginine and histidine. So remember, the non-essential your body can make. Oops, went too fast. The essential you get from your diet. 8 can't be built. You have to get those directly from your diet. And then the absorption and post-absorption states. So here we're looking at lipids, glycogen, and protein. So as a quick review, we have glycogen that needs to be broken down. What can stimulate that is glucagon, that hormone that's made from your pancreas, and epinephrine can stimulate that breakdown. Remember, the red lines are catabolic pathways, blue lines are anabolic pathways. Glucose then gets broken down into pyruvate, lose some CO2, makes some ATP in glycolysis, goes into the citric acid cycle, makes some ATP, lose some carbon dioxide. The NADHs and FADH2 go into the electron transport chain, and we make some energy. Oxygen has to come into this process. We also lose some water. We can have glycogenolysis take place with the help of glucocorticoids and growth hormones to store glycogen up. For the triglycerides, under the stimulation of glucagon, epinephrine, glucocorticoids, or growth hormones, we break that triglyceride down into fatty acid tails, and the glycerol. Glycerol takes the same pathway. It gets converted into pyruvate, and then goes through the steps of the citric acid cycle. The fatty acid tails can be released into the bloodstream. Glucocorticoids stimulate lipid utilization in all tissues except for neural tissues. They work differently, and then you have the blood-brain barrier there. Therefore, there's more glucose available for the brain, but that whole process of going through those chylomicrons and the LDLs and the HDLs, if after they've had those conversions taken place, then those fatty acid tails can go in as acetylcoenzyme A, and we can have beta oxidation take place and give us energy for that. Active liver cells produce so much acetylcoenzyme A that it can be converted into ketone bodies that can be converted back to acetylcoenzyme A by other cells. Proteins, under the stimulation of glucocorticoids and growth hormones, can be broken down into amino acids. The amino acids can be absorbed into the bloodstream by the liver. If we need to break them down, they can go into acetylcoenzyme A, and we can get some energy out of them. So really, this is just a roadmap of everything that we've talked about as far as those 3 different types of macromolecules and where they go into the various stages of cellular respiration, whether it's at the beginning of glycolysis, the end of glycolysis, or going directly into the Krebs cycle. So the absorptive state is a period that's right after a meal when nutrient absorption is underway. So whatever it is that you've eaten, you've digested it down. Now we're going to take those molecules and we're going to absorb them. So the absorption is underway. The post-absorptive state is a period when nutrient absorption is not underway, and as a result, your body is relying on the energy storages and reserves. So the liver cells can break down glucose, can break down lipids, can break down amino acids. So the post-absorptive stage, the absorptive stage right when you've finished a meal, and then the post-absorptive stage is where your body is between meals, I guess you could say, because your body is relying on internal energy storage. There's 3 different types of ketone bodies. Acetoacetate, acetone, beta-hydroxybutyrate. The liver cells don't catabolize or break down these ketone bodies, so peripheral cells can absorb them and then reconvert them to acetocoenzyme A for the citric acid cycle. These acids don't dissociate into solution, so as a result, when you have fasting, it can produce ketosis. It means you have a lot of these ketone bodies are floating around, if you will. Ketoanemia is the appearance of ketone bodies in the bloodstream. Because they are acids, they will lower plasma pH, and then that has to be controlled by various different buffers. Primarily the buffer of bicarbonate has to control that. Ketoacidosis is a really dangerous drop in the pH of the blood. Remember, your blood pH should be at about 7.4, slightly basic, but if you have very high ketone levels, then it's going to cause that to go much lower than 7.4. In severe ketoacidosis, that means the concentration of those ketone bodies is like 200 milligrams, and that pH may go below 7.04. And even 7.05. Even though 7 is still a neutral, your body likes to be at 7.4, which is slightly basic. So even just going down a little bit, too close to 7, it's too acidic, and that can cause your body to go into a coma, it can cause death, it can cause cardiac arrhythmias to take place. So nutrition, some more definitions here: the absorption of nutrients from food. The body's requirement for each nutrient is going to vary. A balanced diet requires all those nutrients needed for homeostasis. Malnutrition is an unhealthy diet, and homeostasis can only be maintained if your digestive tract absorbs enough fluid, organic substances, minerals, and vitamins to meet these cellular demands. All right, so that is the end of metabolism. Next chapter we'll cover is urinary. So I will see you in our next session.